So good morning, and thank you for dialing in this morning. So we'll just give it a few more minutes before we start. And I can see that a lot of participants from Asia has also joined in. So good afternoon to you. We're really pleased that so many of you have signed up for today. And I'll wait a couple of minutes for the rest to join, and then we will start our talk today. Professor Chrisna Gouws.
Should I start or should I give another minute?
We'll give them another minute, and then
Okay. You can hear me fine.
I hear you perfectly. Thank you, Chrisna.
Okay.
Good. It's 2 minutes past now. So before I introduce Chrisna, I would just like to make you aware that any questions you have, please use the Q&A function. And after Chrisna's talk, I will moderate and ask the questions. If there are any questions we don't get to answer, then we'll try to follow up afterwards. Good. So without any further review, I will introduce Professor Chrisna Gouws from Northwestern University in South Africa. So Chrisna, she has been working a lot in establishing 3D models for multi-drug resistance, for instance, but also for drug research in general, including traditional medicine and medicinal plants. So that is what Chrisna is going to talk about today. Chrisna is also one of our dear friends of vivo. So if you want to learn a bit more about Chrisna, you can actually go to our website and look her up under featured friends. So I will hand it over to you, Chrisna, and please let me know if I forgot something.
Hi, everyone. I want to thank Vivo for this wonderful opportunity, and I think it's going to be a very informative webinar series. I will close my video feed to save some bandwidth and try to avoid some lagging. So I'm going to share my screen and end my video. Is that fine?
It's very fine, thank you.
Okay. So as you heard, I'm Chrisna Gouws, and I'm with the Centre of Excellence for Pharmaceutical Sciences at the Northwestern University in South Africa. And I will be telling you a bit more about our use of mini-tumours as models to study anti-cancer properties, specifically of medicinal plants. In my talk, I will focus on 4 main points, including the use of traditional medicines in Africa. Then I'll discuss our motivation for using mini-tumours in our research. Then I'll give a quick summary of the different models we are currently using, and then I'll show you some of the results we have obtained from our anti-cancer studies. So why do we care about herbal medicines and why do we study them specifically? Our university is located in Potchefstroom in the northwest province of South Africa. It's almost in the middle, and that it's about 120 kilometers from Johannesburg. So Africa as a whole, but in particular South Africa, has a very rich biodiversity. We are, for instance, one of the world's 17 mega-diverse nations. And although we account for only about 2% of the world's land surface area, we contain 10% of the world's plant species. So clearly, herbal medicines are then very easily accessible for the people of Africa. And this contributes to the statistics of between 60 and 80% of third world countries' populations relying on traditional medicine for their primary health care. In South Africa alone, there are more than 5,000 plants used traditionally as medicine. African traditional medicine is said to be one of the oldest and most diverse of all medicine systems, but it's very poorly recorded and studied. This is especially important when we consider the use of herbal remedies by cancer patients, as these patients are frequently desperate for a cure, or they frequently suffer from significant side effects when they use chemotherapeutic drugs. They often tend to turn to alternative therapies. The problem is that these often include phytomedicines which are not clinically proven, and it's also used without the knowledge of the treating physician, often in conjunction with their chemotherapeutic agents. As mentioned, plants with medicinal properties from the African continent is largely undocumented and very poorly studied to validate their traditional applications. This leaves us with an untapped treasure vault of therapeutic potential, which we should investigate in depth urgently, especially as cancer is an increasing illness in the world. However, although herbal medicines are usually considered by patients to be safe and without side effects, this is not necessarily true. And the normal drug development pipeline must still be followed when we want to move from traditional medicines to prescribed therapies. Furthermore, it's important to remember when working with plant extracts that it is complex mixtures containing a multitude of components with varying effects. For instance, we see in some extracts both chemotherapeutic or cytotoxic effects and wound healing effects of different components in one mixture. These components can also differ in type and quantity in different extracts based on factors such as the region and the season where they were collected. These variations can complicate standardization of the extracts and therefore the treatments. Another complication in the development of new therapeutic agents from medicinal plants is the lack of physiological relevance of the models mostly used to screen and evaluate these extracts. Traditionally, we use two-dimensional or flat cultures for the in vitro screening, but this is especially for tumors, these do not provide adequate extrapolation to the in vivo situation. This is probably a very large contributing factor to the statistics of only about 3.4% of all new cancer drug candidates eventually having successful clinical trials. And this highlights our need for better models for the screening process. So how do we get these better models that we need? Well, by using advanced technology, we can develop dynamic three-dimensional cell culture models. We specifically culture spheroids or mini-tumours using commercial cancer cell lines, but this technology can just as easily be used to culture patient-derived cells as well, or organoids. In our lab, we first used the first generation clinostat-based system from vivo, the BUM systems, but we have now transitioned to the second generation clinostar system with very great success. You can see it in the bottom corner, what the system looks like. Culturing spheroids in rotating bioreactors in these systems allow high yields of material in a very stable growth environment. The dynamic system allows active diffusion while limiting shear forces, allowing long-term studies of complex models. And what we have experienced as a specific advantage, especially when compared to animal models, is that the system allows for multiple sampling points, which would in animal models have severe ethical restrictions. So why are we specifically interested in mini-tumours or spheroids? There are several cancer model systems available for studying disease pathways and to screen new therapies. And although these models have contributed valuable cancer biology knowledge, they often have major flaws. Two-dimensional cell culture models lack realistic complexity and cell-to-cell and cell-to-extracellular matrix interactions. While animal models are quite costly, very time-consuming, and as you know, have significant ethical concerns. And although I don't think we are close to completely avoiding animal models using these tumors, mini-tumours, we hope to at least restrict and refine the work eventually needed in the animal models. Furthermore, oncology clinical trials have confirmed the necessity of the in vivo tissue architecture to be represented in the models, and the cell extracellular matrix interactions are crucial to effectively screen new treatments. In patients, cancer cells undergo complex limited diffusion of oxygen, nutrients, and signaling molecules, while cellular association, activity, development, and signaling all take place in a highly complex 3D architecture. And it is influenced by the extracellular matrix that cannot be replicated in 2D systems. Our dynamic tumor spheroids show more realistic resistance to treatment, or where metabolic activation of treatment is needed, that is increased. And we have more realistic gene expression and cell proliferation rates in our models. I will now show you examples of the models we are currently using in our group. And I will highlight some of the adaptations to the basic method needed for each cell line, just to show you some examples of optimization sometimes needed when establishing these models. It is important to remember that different cell lines obviously need different approaches, and this can often be guided by the in vivo function of the organ of origin. It gives you some clues of adaptations that may be necessary. Some of the cell lines can very easily self-aggregate to create spheroids, while others need a little bit of assistance to achieve cohesion. And some cell lines just basically refuse. There are already numerous publications for models using the ClinoStar system, and I want to just refer you to a book chapter noted there, published this year, with some of the protocols. And on the Vivo website, you can also find detailed protocols that will give you a very good starting point for these models. So just to go quickly through the basic principles of the methodology used when culturing cells as spheroids using the ClinoStar system, I will just go through the major steps. We normally culture the cells as flat cultures or traditional cultures. And from there, we obtain a single cell suspension. This is usually through trypsinization. And then the single cell suspension must be allowed to aggregate into spheroids. This may happen spontaneously, or it may need some force. So we often use non-adherent plates, and that can be either agrowell plates from stem cell technologies, or we also use spherical plates 5D from Kugelmeyer, or you can use single cell suspensions directly in the bioreactor as well. The spheroids then need time to grow and mature as they form the extracellular matrix and decrease the doubling time of the cells, and also repair any damage they obtain during the single cell suspension processing, like the trypsinization. This allows cells to revert to their original functional purpose rather than only doubling, as is the case in flat cultures. We then fully characterize a new model for at least 25 to 45 days to establish when the model becomes metabolically stable or reaches an equilibrium, which indicates how long the maturation phase for the model needs to be. We look at things like growth rate, protein content, intracellular ATP as a measure of viability, extracellular adenylate kinase to indicate cell death, pH, glucose consumption, and such. After characterization, we are able to identify the optimal experimental window where the spheroids most accurately will represent the in vivo tumor, but this can obviously differ from model to model. Most of our models allow for longer term treatment times than 2D models. In 2D, especially for cancer cell lines that double quite fast, you're often limited to experimental windows of 1 to 4 or 5 days before you have to intervene with trypsinization or some form of subculturing. While with the spheroids, we can often have as long as 2 or 3 weeks experimental time without any dramatic interference with the cells and their growth. So the video there is on the old BUM system. The new bioreactors looks a bit different, but the basic principles are the same. So the first model we used was previously established and published by Versinski and Fay, and it's the HepG2C3A liver cancer model. Multiple papers using this model for various applications have been published. It's a cell line that is derived from the HepG2 hepatocellular carcinoma cell line, And one of the major changes is that the cell line actually has contact inhibition, which the normal HepG2 cell line does not really have. Although the cell line can also be cultured in spheroids from a single cell suspension in the bioreactors, we in our study used non-adherent agarwal plates to create the initial spheroids. And you can see the plate and the, let's call it spheroid seeds on the left. These spheroids were then transferred to the bioreactors after 24 hours, and they were matured in the incubator for at least 18 days before we used them with continuous rotation. Those in the video are already matured. You can see they're already quite big. They start off much, much smaller. The second model we established is the LS180 colorectal cancer model. Colorectal cancer is one of the most prevalent cancers in the world. This cell line we chose because it expresses many of the cytochrome P450 enzymes, and it has previously been used in literature to study chemotherapeutic agents. The approach for this cell line needed to be a bit different because the cell line does not self-aggregate sufficiently to create spheroids. This is one of those stubborn cell lines, so we had to force them a little bit. We encapsulated the single cells in sodium alginate, which is inert and biodegradable. And the alginate was then jellified by cross-linking it with calcium chloride before transferring the spheroids to the bioreactors. You can see we made the droplets on our own non-adherent surface. Clearly, this was a lot more labor intensive, but the student working with it could eventually create 300 spheroids at a time with very little size variation. Dr. Carla Mikalets established this model, and here you can see how the spheroids developed over time in the sodium alginate. And after 28 days, we had quite dense spheroid filling the alginate completely. Ms. Tanya Smith then validated the use of this model specifically for cancer treatment screening. I'm not going to show you all of the data. I don't want to throw too many graphs at you, so I'm just going to highlight some of the graphs. So she studied the reactivity of the model to a standard chemotherapeutic drug. In this instance, it was paclitaxel, which has been prescribed for colorectal cancer. And this way we evaluated the model's potential to be used to screen new chemotherapeutic agents. We used 2 concentrations of paclitaxel, and spheroids were treated for 96 hours with exchange of the media and containing the treatment every 24 hours. Now, yeah, I just want to highlight how we determined the doses and why we do it this way. We determined the IC50 concentrations of the treatment in 2D cultures using traditional MTT assays. We then determined what that dosage was per protein, soluble protein content in each well. In that way, we could convert the IC50 concentration, which was in nanomolar, to an LD50, which was a milligram compound per milligram cellular protein. This allows for easier comparison to in vivo blood plasma levels. So, and then this also allows us to adapt the dosage at every treatment point to the protein content at that time in the bioreactors. So this makes up for any removal of spheroids through sampling, as well as growth of the spheroids. So the treatment is constant per cellular protein present. And then all data was normalized to the untreated control group. In these graphs, it's shown in blue. So although the soluble protein content per spheroid of the paclitaxel treated groups was similar to that of the untreated spheroids, there was a steady reduction in the intracellular ATP, as you can see on the right. Both treatment groups had this effect relative to the untreated control. After 96 hours of exposure, the ATP levels for both treatment groups were reduced to almost half of the untreated groups, with the 2 times IC50 concentration being statistically significantly decreased. On the left, you can see the approximate quantities of glucose consumed by the cells in each treatment group. This was estimated from the glucose disappearance from the culture medium, and we then corrected for the amount of protein present in the bioreactors. It is clear that there was a strong and steady decline with very little glucose consumed after 72 hours. These results indicated that the in vitro LS180 sodium alginate encapsulated spheroid model could be used for future screening for efficacy of chemotherapeutic agents. So this is just a graph of the gene expression of PGP efflux transporter. Paclitaxel is known to be exported from cells via PGP, which is a broad specificity efflux pump protein, which is encoded by the gene MDR1. And it plays a pivotal role in the development of drug resistance. The relative expression of PGP was then measured before and after 96 hours of exposure by means of real-time PCR. Treatment with paclitaxel resulted in an increase in PGP efflux pump gene expression, which correlates with previous studies that showed that paclitaxel induces its own efflux in LS180 cells. The third model I would like to highlight is the H69V small cell lung cancer mini-tumor model. This has not yet been published. We are in the process of publishing it. And this is also currently one of our biggest focus models, which we are going to continue with a lot. We are interested in small cell lung cancer since it has a very poor prognosis and a very high recurrence and drug resistance rate. We are also developing a drug resistant variant of this model at the moment. These spheroids we initiated by placing a single cell suspension directly into the bioreactors and then rotating them very slowly for 72 hours. We also needed to add some ascorbic acid to the culture medium to obtain nice spheroids. This just assists with the extracellular matrix formation. You can see in the top image that we had very round, dense spheroids, and that is a 22-day-old spheroid shown there. You can see it's also quite large. The cell line makes very big spheroids. In the bottom image, you can see a section of a 26-day-old spheroid stained with HE and Alcian blue. Histologically and cytologically, small cell lung cancer is characterized by scant cytoplasm, small nuclei with diffusely distributed and finely stippled chromatin. The cells are small, round, ovoid, and spindle-shaped, and the in vivo tumors reach sizes of between 0.5 and 9 centimeters. The presence of single-cell apoptosis or necrosis and intracytoplasmic mucin in the small cell lung cancer cells are also not uncommon. So in this image of histological staining of our spheroids showed similarity to the typical characteristics of these tumors, and even the presence of mucin in the older spheroids, illustrating the capacity of 3D spheroids to mimic their tissue of origin. So Lizanne did the validation of this model. She used 2 concentrations of irinotecan to evaluate the effects of treatment on the growth and viability of the spheroids after daily treatment for 72 hours. I know in these validations we used very short exposure times, but it was to compare it with the 2D data. So this is not always the time. So after 48 hours of exposure to both concentrations of irinotecan, in this case it was based on the IC25 and the IC50 determined in the 2D cultures, you could see the growth of the spheroids was significantly inhibited, as indicated by a decrease in protein per spheroid, and the spheroids was significantly smaller compared to the untreated control group, as shown in the graph on the left. To evaluate the cytotoxic effects on the spheroids, the intracellular ATP and extracellular AK levels were also evaluated. This is important because the process of apoptosis requires adequate intracellular ATP levels, and if ATP depletion occurs, the cells will still die, but under those circumstances, necrosis would rather occur. So the lower concentration irinotecan decreased ATP per protein after just 24 hours, but this was followed by a peak, possibly due to the cells trying to recover. That's the blue line. The solid black line is the untreated control. I do not show it here, but there was a simultaneous peak in AK release per protein after 48 hours of exposure, which suggested that apoptosis of the cells took place as ATP then markedly decreased after 72 hours of exposure. Both concentrations of irinotecan that were used to treat the spheroids caused a decrease in cell viability, protein content, and spheroid growth. We could also clearly see the appearance of cell debris in the bioreactors after 48 hours of exposure. This is just to show you some of the other models we are currently validating, and we hope to have it published within the next 6 to 8 months. This includes a melanoma model, a squamous cell carcinoma, a breast cancer model, and then, as I mentioned, a drug-resistant version of the small cell lung cancer model we are working with. This just demonstrates that the spheroid culture approach can work for a wide variety of cancer types. And usually we get very nice, round, homogeneous, and dense spheroids. So I will now show you some of the results we have obtained when studying indigenous plants in these various models. As I said, I won't show all the data. You can go and look at the publications if you want more information. Otherwise, it will become a bit overwhelming with all the graphs. So the first plant of interest is Sutherlandia frutescens, also known as cancer bush. It has been reclassified under Lysartia, but we still refer to Sutherlandia as that is also the name it is commercially sold under. It's widely available. Even in our pharmacies, you can buy this plant material to create teas. So it's very easy to obtain. It typically grows on the west coast of South Africa, but it has now also become a general garden plant in many regions of the country. It has a very long history of traditional use for cancer treatment, hence the name cancer bush. And although it is more recently been prescribed as an immune booster, specifically in TB patients. The typical application is ingestion of a tea or an infusion of the leaves, the dried leaf material. So in our studies, we also prepared a crude aqueous extract to use. Important with this plant is that several studies have confirmed that no physiological toxicity is associated with the use of cancer bush, and this includes a clinical trial where they gave patients quite high concentrations of this extract with no adverse effects. So we investigated the effect of 96 hours exposure to 2 concentrations of Sutherlandia, which we replaced every 24 hours, specifically on the growth and viability of 12-day-old LS180 spheroids. So this would give us an indication of applicability in colorectal cancer. We replace the extract every 24 hours as plant material are notoriously prone to decomposition or breakdown of some of the components. So to avoid that, we replaced it frequently. The patients also typically drink the tea at least once or twice a day. As seen in the graph on the left, the intracellular ATP per protein was decreased significantly after only 24 hours of exposure. These undetectable levels of ATP remained as such for the duration of the experiment for both treatment groups. That's the blue and the orange lines, when compared to the untreated control, which is the solid black line. The ATP content decreased much faster and was much lower than that of the group treated with paclitaxel, which is the dotted black line. This significant depletion of intracellular ATP was indicative of a loss of cell viability. The normalized glucose consumption shown on the right also decreased following treatment with Sutherlandia. The decrease in glucose consumption supported the conclusion that cell viability was decreased following treatment with Sutherlandia. So here I would frequently now get a question about concentration, especially when we compare the plant extract to a chemotherapeutic drug. Direct comparison is very difficult because with the extract, as we mentioned, you have a very complex mixture of numerous components, while the reference drug is a single active compound. So we rather look at the tendency or the tendencies that visible and the reactivity of the model to directly compare the concentrations. We would need to eventually look at fractions or isolated compounds from the extract we prepared. In another study, we treated 16-day-old small cell lung cancer spheroids for 96 hours, again with the crude aqueous extract of the Sutherlandia. And again, the treatment was repeated every 24 hours. In the graphs, green represents the IC25 determined in 2D, the orange IC50 and the red IC75. The black line is the untreated control and the dashed line represents irinotecan. I think that was also IC50 of irinotecan. So Sutherlandia caused a concentration-dependent loss of viability of the spheroids, as indicated by the decrease of intracellular ATP levels shown on the left there. On the right, there was a significant increase in AK per protein, which I will remind you it's adenylate kinase, which is a marker of cell death. The AK of the steroids treated with IC25 remained elevated for the rest of the treatment period, while after 72 hours exposure, the AK content for both the IC50 and IC75 groups decreased and was below detection limits. The increase in both ATP and AK caused by the IC25 group of Sutherlandia indicates cell death, most likely due to apoptosis. In contrast, the reduction of ATP and AK to below detectable limits after exposure to the IC50 and IC75 groups after 72 hours exposure also represents cell death, but in this case it was most likely due to necrosis of the cells. We observed clearly that cell death following treatment with Sutherlandia was both concentration and time dependent. So second plant of interest is the Smalobium undulatum, also known as Uzara. It's one of the most widely used phytomedicines in South Africa and is known to contain plant cardenolides, which have emerged as promising new cancer treatment agents. Although it's not clear if this is a cancer-specific activity or if it's just general cytotoxicity. So in a subchronic study in our liver cancer model, we studied potential cytotoxicity of the plant, and there was clear growth inhibition following treatment with two different concentrations. Those are the two lines in the middle, while the top line is the untreated control and the bottom line is the positive control. On the left, the intracellular ATP levels, and on the right, the extracellular AK levels also indicated a loss of cell viability. Although it seems that the cells coped with the stress of the extended exposure. It wasn't such a dramatic effect. When we treated our colorectal model with the Uzara, we also treated 12-day-old spheroids with IC50 and double the IC50 concentrations determined in the 2D model. again, we could see a steady decrease in the intracellular ATP content, which is the blue and the orange lines on the left. Although this decrease was only significant after 48 hours of exposure. Relative to the paclitaxel group shown in the dashed line, the ATP decreased significantly and much faster than for the paclitaxel. On the right, the glucose consumption also decreased steadily following treatment, and that was very similar to the effect the paclitaxel group had. And therefore, we could conclude that Uzora does have anti-cancer potential when considering the decrease in cell growth and viability. But again, safety studies are needed to confirm that there's no general toxicity. So to conclude, it's clear that the various mini-tumor models are reactive to treat cancer treatments, and they are suitable to study new anti-cancer treatment options. Sutherlandia was shown to have notable activity in both the colorectal and small cell lung cancer models, and we will study this now more extensively, especially as it's known to be non-toxic at high concentrations. We will now move to fractions and then eventually maybe to single compounds, although my gut feeling is that it is a synergistic effect of multiple components. But this result supports the traditional application of this extract to treat tumors. Uzora did have some activity, although not as pronounced as Sutherlandia, and as it has a higher toxicity risk, it's probably not an ideal candidate unless we can identify a single compound that's active but non-toxic. Here are just a list of the references used. Then I would like to thank my funders, the South African National Research Foundation and Medical Research Council. And I would specifically like to thank Vivo for being my partner in research for a very long time now. I would also like to acknowledge my team that has worked on this. Apologies, Dr. Hanna Svetina, I did not put it here. She's back in the Ukraine. And Dr. Karla Mikalets is no longer in Sweden. She's now in the Netherlands. But these hard workers made all these research results possible. Thank you very much for your time and your attention. And then you're welcome to go to the questions.
Good. Thank you very much, Chrisna. Thank you for a very, very interesting talk. I'm super excited and also very excited to see the new results coming out. Yeah, so thank you very much for giving this talk. We have quite a lot of questions coming in, so I'll try to divide them a bit. First, I'll talk a bit about, ask a bit about the use of the model systems. So I have one from Australia who asked, do we have to strip away the alginate prior to Laxetal treatment?
No, we did not remove the alginate. The alginate has been shown to allow diffusion of compounds through it. And it has previously been used by other researchers, which has shown that eventually it is degraded and removed naturally by the as the cells grow. So, but the alginate does allow diffusion of treatments. So, no, we did not remove it.
Okay, Good. Thank you very much. I have another question. Says, nice talk. I'm wondering what is the effect of the rounds per minute and the starting size and the quality of your spheroids?
Sorry, just the RPMs.
Yes.
Okay, so every cell line needs some optimization. So we have seen that you start with the smallest amount of cells you can, and because you don't want overly big spheroids, because the faster they become big, the shorter your experimental window will be, because you will get to a point where diffusion rates becomes a problem, and especially for metastatic, highly metastatic cells, you get a problem if they are too big. So you optimize per cell line. Obviously, you want to have spheroids that is a workable size. So the single cell suspension, we optimize to see where we get nice spheroids with a good yield of spheroids without too much debris. While in the plates, we start with will easily see in the plates, if it's too few cells, they won't make nice shapes. If you have too many cells, they overgrow and you get like multiple spheroid strings. You don't get individual spheroids. The rotation speed, the RPMs, if you start with a single cell suspension in the bioreactor, you start as slow as possible. So I think we start with 2 or 3 RPMs, because you do not want to have too much motion, so the cells don't have time to aggregate, and the velocity will just pull them apart. While if you have pre-formed spheroids you place in the bioreactor, you want to optimize the speed. As the video I showed, you want to have them have as little motion of the spheroids as possible. You want to have them kind of static. So if it's too slow, you will see them drop in the bioreactor. If it's too fast, they will spin out and hit the sides of the reactor, which will damage them. So it's very much a look and feel thing. There's not a specific rotation speed necessarily. It's optimized per cell line, and you adjust it every day to adjust for the growth of the spheroid. I hope that answered the question.
I think it was a very nice answer. Thank you, Chrisna. I have one more with regards to the spheroids in the bioreactor. So it's what are the sizes of the spheroids in your bioreactors, and how many can you have in your bioreactor?
So that again depends on the cell line. The nasal cancer line that we now are busy with are very, very small. They are 200 to 300 micrometers in 200 micrometers in diameter, and they don't really increase in size over time. They kind of only become more dense. So even after 3 weeks, they are still less than 500 microns in diameter. Other cell lines increase in size very fast. For instance, our melanoma cell line, they become extremely big. So after 20 days, 25 days, you have more than 1 millimeter in diameter. So it varies quite a lot, and that's why I say we try to start as small as possible, so that they don't become too big too fast. It depends on your downstream applications how big you want them also. For microscopy, we prefer them being a bit smaller. So the number of spheroids you can have in each bioreactor also then depends on the size and each cell line. We optimize this. We do a pre-characterization where we see where the spheroids start deteriorating in terms of viability. So we check ATP and protein content and pH and glucose consumption. And that indicates to us when we have to split our population. So that shows us when the population is too high in terms of the size they are. So for one cell line, we will need to divide the population on day 8. In another cell line, we can divide them only on day 12 or 15. But typically, you can start with around 300 spheroids per bioreactor. We have had cell lines that, especially with the sodium alginate, you want the population a lot less dense. So I think there we worked with about 150 spheroids per bioreactor at the beginning. So it depends on the cell line and their behavior, their glucose consumption. We have one cell line that very quickly lowers the pH of the culture medium. So there we have to make the population a lot less dense. But the fewer spheroids you have, the faster they grow. So it's again, you must optimize for each cell line what is the ideal density at each time point in the culture.
Thank you very much, Chrisna. I think I have 2 more questions with regards to the procedure of using the system. So one is, how does the ascorbic acid help spheroid formation? Is the ascorbic acid only applicable for the TH69V, or can be used for other cell lines or primary cells?
Okay, so we have used it for different cell lines. It's one of the steps when we have trouble or getting very nice compact spheroids. It's one of the additives we try and see if it helps. What it does is it assists in the process of moving the proteins from the inside of the cells to the outside of the cells. So it doesn't increase production of the extracellular matrix, but it assists with the excretion of the material, if that makes sense. So some of the cells have trouble with that process in vitro. So that's one of the things you can try to assist in the extracellular matrix forming nicely and getting nice dense spheroids.
Okay, thank you very much. Then I have the final question with regards to the system is, have you encountered the problem that spheroid aggregate in the bioreactors?
Yes, that's a frequent problem. Again, there's quite a few different things you can do to avoid it. We have in the past played around with coating the spheroids or treating them with EDTA or coating them with serum before transferring them to the bioreactors. That's some of the things you can try. What we do see is that aggregation happens specifically in a couple of cases. The first thing is if you do not wash the reactors very well beforehand, you can have some production fibers in the bioreactor. So it's very important to wash them very well before you start your culture, because those fibers or foreign materials will grab the spheroids and then you get aggregates. Then also if your spheroids is not smooth, especially for the like our sodium alginate model, the colorectal cancer model, it's a very highly metastatic model. So when the culture becomes too old, we saw that the cells kind of started to climb out of the alginate, and then those protrusions will also grab each other and you get aggregates. Some cells just make the aggregates easier than others. It's important to then work with your spheroid density in the bioreactor. If it's too dense, then you will get these aggregates easier. But we do have one cell line that we see sometimes it starts to make aggregates, but if you just disperse it, they continue again without major damage. So there's a couple of things you can play with, but yes, for very stubborn ones, you can try coating them with FPS or treating them with EDTA before placing them in the bioreactors.
Okay, thank you very much, Chrisna. Then I have a question here called, have you used essential oils or supercritical extracts in earlier mentioned assays?
Sorry, just repeat that.
Have you used essential oils?
Oh, well, essential oils.
Yeah.
No, I have not used it. I think with oils, you may have a problem of getting it dispersed properly in the media. It may float. So if you can get around that problem, maybe, what's the word now? Make an emulsion or something. It could be possible to use it. We have not tried yet, no. One of our biggest challenges when working with the plant extracts is to make sure that there's not too much plant material debris left, as this will again increase aggregation of the spheroids.
Thank you very much, Chrisna. So, nice answer. And the final answer, final question, we are past 10 o'clock now, is great discussions. In conventional cancer studies, there is usually the need to determine the selectivity indices of tested samples to determine selective cancer toxicity. Is this the case in these studies? Or is it relevant to using the 3D technology?
There's two sides to the coin. So the first, yes, we are always interested in the selectivity index. That's why we are not really continuing with the Uzora, because it has a very low selectivity index, while Sutherlandia is known to be not toxic. So on the one hand, we do the selectivity index just in our 2D MTT screens as an indication. So that gives us an indication of the selectivity index. On the other hand, yes, we would prefer to do the selectivity index in our 3D models as well. The problem there is that you need non-cancerous cell lines, which is always a challenge. So we do now have a lung fibroblast line that we want to culture as spheroids, which we would then like to include in our panel. So we'll have a non-cancerous line, drug-sensitive small cell lung cancer line, and then a drug-resistant small cell lung cancer line. So that's the direction we're moving into, and we hope to get there. But currently, we are not doing it in the 3D model now.
Okay, good. Thank you very much, Chrisna, for a very nice answer. Then I think I will end our webinar today. I'll do that by thanking you a lot, Chrisna. I was I learned a lot today, and I'm very pleased to see all your great results. So thank you once again, Chrisna, for a very good talk, very nice talk. And thank you for our collaboration. We value it, I guess, minimum as much as you do. And then I would like to all our listeners out there, please sign up for or see the rest of the webinar series which we have on the way. So make this. We have the coming Thursdays. We have new webinars. So we have Steve, our founders, next Thursday. Simone Chidoli, October 7th, and then we have Adelina on October 14th. Thank you very much for listening, and I wish you a great rest of the day.
I want to thank you for the opportunity. I will also now just put my email address in the chat box. If anybody wants more information or wants to talk about the information, the presentation, you're welcome to just email me.
Thank you.
Thank you.